All right. Hey, everybody. Dr. O'Halloran is a plant ecologist with an interest in how plants respond to changing environments at different spatial and temporal scales. She holds a bachelor's in biology from Oberlin College and a master's and Ph.D. degrees in environmental sciences from the University of Virginia. She received a postdoctoral training from Dr. Lee Hanna of Conservation International at the University of Integrative Biology. At the University of California, Santa Barbara. She continued her postdoctoral training with the NSF-funded Nutrient Network in the Department of Integrative Biology at Oregon State University. She started teaching Ecology and Conservation Biology in the new online undergraduate program at OSU. Following that, she returned to Central Virginia, where she continued to teach ecology and conservation biology online for OSU and became a visiting assistant professor at Sweet Briar College in their Department of Biology in 2015. In 2017, she started teaching for the online master program in the Department of Forestry and Environmental Conservation at Clemson University. Dr. O'Halloran's research interests are in plant ecology, global environmental change. As plants are faced with novel abiotic conditions, they are forced to adapt, shift their range, or die. She examines how plants respond to environmental impacts at the spatial scales, for example, loss of fitness or phenotypic plasticity, and at ecosystem scales, for example, changes in gross primary productivity, relative diversity, and abundance, in hopes of providing the science necessary for climate change conservation planning. So with that, I'll turn it over to Dr. O'Halloran.
Great. Thank you, Jessica. And thank you, Thea, both of you, for having me today as your seminar speaker. Thank you for coming to my talk entitled Plants in a Changing Environment, How to Study the Future. So studying the future might sound like a daunting task, and it is. However, plants do give us lots of clues as to how they may or may not survive in the future. So my job as a plant ecologist is to help uncover these clues to enable us to understand how we should expect plants to respond to global change. Global change caused by a differing climate and land use change and consequential alterations to global nutrient cycling. So plant response to global change is intricately determined by ecosystem structure. And ecosystem structure describes the abiotic and biotic conditions in a given space at a given time period. The relative abundance of producers, consumers, and decomposers, and how they use these abiotic resources determine the processes and ultimately the ecosystem structure. So the main drivers in the climate cycle that affect the resource availability and ecosystem structure are precipitation and temperature. The sixth assessment report published by the IPCC came out in February of 2022. Working Group 2 focuses on the impacts, adaptation, and vulnerability of Earth's ecosystems. So in this report that was recently published and accepted was that unless we curb our emissions, that we are committing to a certain amount of temperature change, the average change for global temperature. So in this figure from the IPCC report, it just says by 2100, unless we level off our emissions as indicated in the green line, we're committing ourselves to this higher temperature for the Earth. So the impacts of this type of temperature change to current ecosystems specifically is unknown, but a lot of the recent literature gives us an idea of some of the general patterns that we've noticed. My computer is frozen a little bit. Okay. The sixth IPCC report that I mentioned basically is a collection and distillation of hundreds of published research that has been published since the last report, which was in 2014. So they're not actually conducting any new research, but more of a meta-analysis. I want to focus on, in this figure on the far left, the area that's in the red box. The first column that we see are different geographic regions, and then closer to the bottom, I don't think my pointer is going to work on here, is our areas broken down by Earth's major biomes. And this figure shows the confidence in which we can attribute major ecosystem structure changes to climate change. So where you see the different arrows pointing, this is broken down into terrestrial systems, freshwater systems, and ocean. The darker circles indicate very high confidence in attributing climate change, and the lighter circles, or the empty circles, indicate that there is not sufficient evidence. So as you can see, for terrestrial systems, it's pretty high confidence that climate change is impacting these different areas. And this is the areas in which I work are terrestrial systems. So that's kind of what my focus is going to be on. The report does also say that there is evidence of major species range shifts. And for those of you who have taken some of my classes, we talk about this. Major responses of range shifts being species moving to higher latitudes, but then also to higher elevations for those montane species. And then another major impact is that of phenology, so changing of biological events, things like migration, peak biomass productivity, reproduction, any type of events that are driven by temperature cues can be impacted by climate change. And again, we see the darker circles, major impacts for terrestrial systems, because that's where a lot of the research has concentrated. So how will plants respond to affect this ecosystem structure? One mechanism that I believe that will enable plants to respond is their ability to adapt to changes in short-term environmental stochasticity. So things like droughts that we're seeing more of, increased nighttime temperatures, changing in fire frequency. These are all new environmental stochastic events that are driving ecosystem structure changes. So just journey with me for 1 minute to 3 different ecosystems that are characterized by the presence of both woody tree species and herbaceous species. On the top panel, number 1, we see a salt marsh in South Carolina characterized by cordgrass and needlerush juncus. And then on the horizon, we can see some live oaks and longleaf pines. Excuse me. This ecosystem experiences a lot of saltwater inundation from high tide events, king tides, annual, just seasonal storms, but then also more rare events such as hurricanes. In panel number 2, there we see an ephemeral wetland in central Virginia, and this is characterized by rapid changing soil moisture throughout the growing season from precipitation events. On the far right is, in panel 3, is the eastern slope of the Sierra Nevada mountains, where species such as Jeffrey pine, pictured here, and lodgepole pine, depend heavily on the soil moisture from snowpack. And if you look way in the distance, you can see the last remaining bit of snow. I took this picture in June, I believe. And so that's really the kind of the remainder of the snowpack that's providing the soil moisture. So what do all of the plants in these environments have in common? Well, they're susceptible to very highly variable environmental conditions. And so the ephemeral wetlands are changing on a daily, monthly, yearly basis. The precipitation pulse events from snowmelt allow pulses of nutrients. And so all these plants can thrive, but how have they adapted to these conditions? So broadly, I'm interested to look at these adaptations And especially in the face of climate change, are there some species or taxonomic groups or functional types, maybe even populations of a given species, that are more able to express phenotypic plasticity than others? So phenotypic plasticity is the ability of an individual to express different forms of a gene in response to its environment. And this can be found in all different types of organisms. So examples include, and some of these you probably have seen, cephalopod color changes, the sex reversal of goby fish, changes in root to shoot ratios for crop species depending on nutrient availability, stomatal size and density on the undersides of leaves, and leaf thickness for shade or light leaves in a given within a canopy. So all of these changes occur within an individual's lifetime. And usually these changes are advantageous, meaning that they result in an increase of survival or fitness for that individual. The changes that occur are not necessarily inherited by the offspring, even though the ability to express these changes might be. So the exact mechanisms of what allows individuals to be more phenotypically plastic than others is not thoroughly understood. I'm going to talk a little bit now about some of the research that I've done examining this. And this research took place in the central Virginia wetlands of the picture that I mentioned earlier. We used a model species, Mimulus ringens, which produces this kind of purple snapdragon type looking flower. It's an herbaceous perennial found in wetlands, and there's also analogous species that are found on the West Coast. It exhibits a mixed mating system, meaning that it can reproduce by outcrossing, but also selfing, and is pollinated by bombus. It can also reproduce vegetatively underground through the production of rhizomes. So we're interested in looking at phenotypic plasticity as a mechanism for adapting to change by using 3 different soil moisture types, low, medium, and high, that changed, that are ephemeral, so it changed throughout the growing season. And then we looked at both outcrossed and selfed individuals to see if there was potential inbreeding effects. So as populations become more constricted or bottlenecked because of human encroachment, it's a possibility that there may be increased levels of inbreeding. In order to get an understanding of how much plasticity there was, we looked at both sex-linked and non-sex-linked traits, anything that might be resulting in an increase of fitness. So first flowering date, total flower production, corolla, hercogamy, which is the spatial separation of anther and stigma. This is getting into some really detailed botany, but the hercogamy is, will come up again in a minute. So, and then we looked at some pollen and growth rate, and different photosynthetic qualities, stomatal resistance, and carbon assimilation. And in general, we found that some of the inbred plants did have reduced hercogamy. So that, remember, that means a decrease in the space between the male and the female parts, which could, over time, if this population is still restricted in its amount of outbreeding, that it could result in selfing over generations, kind of exasperating the effect of inbreeding. We found that those plants with medium amounts of soil moisture were the happiest. Basically, they were growing the fastest and produced the biggest biomass. And that in general, plasticity was not affected by inbreeding, but there were some, except for some traits. And so it's possible that maybe these populations, as this was only one generation of inbreeding, it's possible that over time these deleterious alleles would accumulate. So moving on to another study that is kind of looking at, again, changes in environmental conditions to understand better how vegetation responds and has evolved. This study took place in Southern Africa in the Kalahari region, and what makes this area really unique is the presence of relatively undisturbed homogeneous Kalahari sands. So the orange part in the middle, you can see throughout the middle of Africa, indicates the presence of savannas. So again, this coexistence of tree and grasses as a common vegetation type. The Kalahari transect was identified because of the common soil type, but along this very dramatic precipitation gradient, which then of course gave rise to a vegetation gradient. It has been identified as an international biosphere site because of this very unique combination of vegetation and soil along this transect. So we identified 5 different sites where we were looking at the vegetation and nutrient controls. So in the most northern part, the canopy is about, I'd say, 60% closed with woody species. And as you move further south, the canopy begins to open up. And throughout all of these sites, there are lots of grazers and browsers and different carnivores, making it a pretty exciting and sometimes dangerous but fun place to conduct field research. So throughout the transect, we set up experiments where we were looking at increased nutrients along the precipitation gradient to see how that would change productivity and ecosystem structure. We looked at soil nitrogen and phosphorus, and then also took samples throughout the wet and dry seasons of the foliar tissues to look at nutrient concentrations, and also looked at productivity through biomass, and then also some photosynthetic responses, which I didn't here, but looked at photosynthesis and carbon assimilation. An interesting thing that we found was that there was very high amounts of interannual variability of production, of biomass production across the transect. And this probably is an indicator of the amount of interannual rainfall that we see. So even within the transect, there is rainfall variation from year to year. We saw evidence that phosphorus was being taken up by the vegetation, but it wasn't always evident didn't always translate to changes in biomass. So that brought up a lot more questions of what is happening to the nutrients, the phosphorus in particular, and how this is being used and stored possibly below ground, or used or stored in leaves, but not necessarily used for biomass productivity. And then of course, it's brought up a lot more questions that still need to be answered. Similarly, this study was expanded to a kind of a larger scale nutrient network study. And this was what Thea mentioned in the introduction, where we're trying to address some of these same questions of nutrient control for ecosystem structure, but at a global scale. So using a coordinated effort of over 50 scientists to identify or to use identical streamlined methodologies so that we can compare results without having conflicts by using different methods. And so all of the sites are using sampling techniques and biomass harvesting, carbon assimilation relationships to kind of understand more of the productivity and diversity relationships across a global scale. And a lot of these data are available, publicly available, which makes the data set really nice because it's so comprehensive and kind of user-friendly. So the main questions that the the network is trying to address are how general are our current understandings of productivity and diversity relationships across the globe? To what extent are plant production and diversity co-limited by nutrients for these herbaceous dominated communities? And under what conditions do grazers or fertilization, in other words, eutrophication, control plant biomass diversity and composition? I'm not going to go into too many details of results right now, but I just wanted to highlight how this project is a great opportunity for graduate students and undergraduates. There's lots of RU projects that have spun off of this. They're great teaching opportunities, and there have been lots of publications that are continuing to come out from this project. All right, so now I'm going to talk a little bit more about my research at the Baruch Institute. So I would like, if I could see all of you, I'd be asking how many of you have actually ever been to the Baruch Institute? It's located pretty far from campus. Since Clemson is home to, or is a, oops, did I freeze here? Okay. Clemson is home to 6 research and education centers that are found throughout the state, and the Baruch Institute is one of them, and it's specifically designated to encompass research and teaching of the coastal ecosystems, as well as other ecosystems that are representatives of South Carolina kind of as a whole. It's about a 4.5 to 5 hour drive from main campus. And as you can see in this zoomed in Google Earth image, the Baruch Institute is located on the Waccamaw Neck. It's a peninsula that's surrounded by, on one side, the Atlantic Ocean, and then the other is the Winyah Bay and the Waccamaw River. The Baruch Institute has lots of facilities that are available for faculty, research, and undergraduates. It is located on Hobcaw Barony, which is a designated conservation area encompassing about 16,000 acres. And what makes it unique is that it's surrounded by areas that are very developed. So if you've ever been to Charleston or Myrtle Beach area, these are all, highly developed areas that are in the larger biome of the North Atlantic coastal plain. And so the Baruch Institute at Hobcaw Barony really is kind of in the midst of this island of conservation area. The facilities include lots of office and lab spaces for water quality and different environmental research. There's a conference center and education facility that's used for summer classes, office, smart room, and meeting rooms. And then there's also lots of summer housing that can be used by undergraduates or graduates that are conducting research. Like I said, it's a fairly unique place to work because of the diversity of ecosystems here. So because we're surrounded by the Atlantic Ocean and different estuarine habitats and the freshwater river system, there are lots of different opportunities for comparative studies. So there's coastal forested ecosystems, riparian, wetland ecosystems. There's also further inland longleaf and loblolly pine forests that have different management regimes, making them kind of a natural template of investigations of different clear-cut, managed fire habitats, and some older growth longleaf and loblolly pine as well. It's a birder's paradise. They do the Christmas bird count here every year. Lots of interesting species that have motivated different conservation researchers to come. At Hobcaw, my personal research interests are kind of broad scale plants responding to global changes. So More specifically, I'll go into some more specific examples in a minute, but just as a whole, kind of investigating how plants will and what mechanisms they will use to respond to global change. So how can we ultimately develop appropriate and effective conservation strategies to ensure that the species that are important to South Carolina as natural resources, but then also as endangered species, how can we help protect those through different conservation regimes? So when I say protect, what exactly am I talking about? Well, this can be looked at from both the species scale and ecosystem scale. At the species scale, I'm interested in maintaining effective population sizes. So kind of trying to understand what population sizes are necessary to maintain enough genetic diversity so that we don't have inbreeding, like I'd mentioned in some of the previous mimulus research. And then from an ecosystem scale, what is the appropriate habitat necessary, that the ecosystem structure that will support these species? The natural resources that we are focusing on in this lab are loblolly pine as an important South Carolina timber species, the longleaf pine ecosystem as a whole because of its kind of fragile balance that has been managed by forest and, managed by fire and control burns, and then also some rare coastal herbaceous species. So I'm going to go into each of those in a little bit more detail. Loblolly pine, like I said, is an important timber species, as we all know, for South Carolina. And there are certain parts of the loblolly pine kind of development from the nursery scale out into the planting time period that this species may be more susceptible to changing environmental conditions. So especially now as we are seeing prolonged periods of drought and colder or warmer nighttime temperatures, we're interested in seeing, are there different kind of thresholds in which these, especially in the seedling and young stages, that the species can survive. Longleaf pine is also an important coastal plain ecosystem. And so I'm interested in investigating how these ecosystems exhibit differences in recruitment, for example, in wet and dry years. So we are seeing a lot of interannual variability in the timing of precipitation events. And especially after years in which there's been prescribed burning, there might be different nutrient pulses based on the timing of these precipitation events. So is there an upper threshold for growth in these periods of extreme drought? And then are the areas that have been labeled as conservation areas account for range shifts that may be induced by climate change? Because these longleaf pine ecosystems currently occur in these different pockets, are we accounting for the possible movement of them to higher latitudes? A unique aspect of the Brook Institute is its proximity to a lot of the beach environments up and down the Carolina coast. So if you've ever have been to this area, you probably have seen that most of the coastline is developed either with commercial areas or residential communities. So the Brook Institute is closely situated to Huntington Beach State Park, which is a protected area where there is no development, as well as the adjacent Brook Green Gardens. And this area has started to be identified as an area to study a threatened herbaceous species, the sea beach amaranth. And so I'm interested in kind of trying to understand better how this plant will respond. What are the saltwater inundation thresholds to sea beach amaranth? And specifically, how does this affect the survival and fecundity for the species? Is population augmentation a feasible conservation method? So for green gardens is one of the kind of leading conservation botanic gardens for this part of the state. And so trying to work with them on figuring out if we can provide individuals based on a specific gene pool to be planted in the coastal environments. And then, do managed populations in captive settings exhibit the same genetic diversity as the wild populations? A project that has just been started this growing season is very similar to the nutrient network study that I was describing earlier. I am specifically interested in how soil nutrients and disturbance impact ecosystem scale processes related to plant growth and decomposition in this an old field site surrounded by longleaf and loblolly pine forests. This is will be part of a global study, so contribute to a data set that can be used to compare across different similarly herbaceous dominated ecosystems. So using eutrophication or increased nitrogen and phosphorus and micronutrients, as well as different disturbance treatments, I'll be looking at plant productivity, community composition, seed bank dynamics, and also native versus invasive plants to understand kind of how species composition is related to productivity. So this study is, and I'll mention a little bit more in a minute, we're looking to expand our graduate and undergraduate involvement for this. And so if you are interested in possibly applying for undergraduate research, there are different opportunities to become involved in this project, as well as for graduate research. So some of you, I was just looking through the names of some of you in attendance today. I think I have come across in my teaching. I teach for the online program, and I am currently teaching 2 classes in the fall and the spring: global change ecology and advanced conservation biology. So I think I recognize some of the names, even though I haven't met all of you in person. And like I said, we are recruiting students who may be interested in working in some of these ecosystems. If you're interested in joining the lab, I encourage you to check out or to email me or check out our website that has information about potential PhD students. So if you're currently a master's student or thinking about a PhD, please reach out if some of these topics are of interest to you. You can also check out Twitter that has a link to the website, I believe. So let's see, yeah, so I'm happy to take any questions or to discuss any of the topics or research that I touched upon. And then I know we said we could also have more informal conversation as well.
If anyone has any questions, they can type them in the chat. I'll have that open and can moderate that, or you can unmute yourself or raise your hand and ask a question. Or I could start asking questions if no one else has any. Here is the first one in the chat box. I'm wondering if you not only observe, but also model phenotypic plasticity.
Oh, that's a good question. I have not modeled phenotypic plasticity, but a good question would be, can you model it also? And probably you could. I have not done that, but it's definitely something that could be interesting because there are a lot of data that could be used to kind of validate and verify a model based on previous observations and just the ability to easily manipulate environmental conditions. So usually phenotypic plasticity are the data are kind of described by what they call reaction norms. And so so reaction norms are basically a figure that show maybe 2 or 3 modes of a environmental condition. So you can think of like the low, medium, and high water, and then basically looking at how the whatever trait you're looking at kind of manifests. So the more previously in phenotypic plasticity research, it's been a kind of a bimodal reaction norm, but in fact, I think research is showing that if you look at more, making it not just a bimodal kind of linear relationship, but trying to figure out if there's more nuanced responses that you might miss with a linear response, that would be a better model for trying to describe the plasticity. That helps a little bit.
Okay, he says thank you.
You're welcome.
So I think you talked a lot about, and feel free to type your question or unmute yourself if any of the students have questions, but you talked a lot about the use, these different characteristics of plants that you're studying, their use for plant conservation in the face of global climate change. But I do a lot of work on restoration or, trying to bring ecosystems back after they've been impacted. And so I think a lot of the questions are similar or overlap. So a student, if she hasn't dropped out, who was here, Ellie, who graduated not long ago, looked at little bluestem and particularly locally adapted phenotypes and how those survived much better, we found, in the Sandhills region of South Carolina than seeds that we ordered offline for little bluestem, which can occur really anywhere in the North American continent. So I think that there are potentially some additional applications for this work when we talk about really restoring systems that have already been infected.
Oh, yeah, absolutely. Especially when you are looking at, regional kind of like either microclimates or regional drivers of certain conditions that might more likely support the specific genetic variety of an individual. And so that of course applies also to managed systems like in agriculture, just specific crops that might be more adapted to higher salinities that are grown along the coast, things like that.
Yeah, I think in particular in that Sandhills region of South Carolina where she did her research just outside of Camden, it's drought tolerant. So they were putting a lot more energy into the below ground roots and growing them a lot longer than the seeds we ordered online as a way to survive that. So thinking about that moving forward, you could also use this to plan for an uncertain future or predicted decrease in precipitation or increase in drought.
Right. Yeah. And just maintaining, I think, I don't know how much of a kind of genetic safety net we have, for crops and plants in South Carolina, but to be able to kind of store that genetic diversity so that we can enable, restoration projects where we might need different genotypes.
Okay, we have another question in the chat from Pravina. Can we use phenotypic plasticity to explain invasiveness of species?
That's, yeah, also another good question. And that it kind of touches on this idea of that I recently have read about of just, the expression of phenotypic plasticity in R and K selected species, or certain species that have given certain life history traits kind of more able to capitalize on resource availability. So invasives frequently, have the ability to exist and compete with other species because they can access resources either earlier in the growing season so that they're, using the early light availability or nutrients. And so, yeah, I think that it would be interesting to look at kind of the relative expression of phenotypic plasticity for invasives versus, natives.
Yeah, or even thinking about, I think it would be most interesting to compare invasives to some of our sort of ruderal, like dog fennel or something ruderal that is native, but similarly will quickly invade or take over a site and spread. Are there any other questions out there? Okay, I think I'm going to stop the recording and